So now we're going to take a look at the complexes that are in the electron transport chain. Four electron transport complexes are independent of one another. There's no evidence that they associate with one another in the membrane. However, three of them contribute to the proton gradient that drives protein synthesis. And you'll see that complex 1 pumps protons to the intermembrane space, as does 3 and 4. If you take a close look at complex 2, you'll notice that it's actually an enzyme from the citric acid cycle, where we have succinate being converted to fumarate. Remember, the other enzymes for TCA are in the mitochondrial matrix. So we just talked about standard reduction potentials, and the standard reduction potential for molecular oxygen to water is 0.815 volts. That's positive. So that means it's a very good oxidizing agent. And actually, it's the most effective oxidizing agent than any other biological molecule. If NADH were directly oxidized by O2, there would be a large release of free energy that could not be utilized by the cell. Thus, the evolution of the electron transport chain. We've taken a look at some of the molecules involved. We have flavoproteins, we have FMN, and we have FAD. These are one or two electron transfer agents. We have, which you haven't been introduced to, CoQ, and this is coenzyme Q, or ubiquinone. It can transfer one or two electrons as well. We have cytochromes that contain heme groups. These are one electron transfer agents where Fe2+ is oxidized to Fe3+. We will also see iron-sulfur proteins, where we have a one electron transfer. And again, this is the oxidation of iron 2+ to iron 3+. And finally, we'll see in complex 4, a copper ion that is able to transfer one electron. The electron transport chain and ATP synthesis cannot occur without the other. These are coupled processes. There are no regulators of the electron transport chain other than the lack of oxygen. So let's take a look at complex 1. This is the largest of the electron transport proteins. It has 46 subunits. It weighs about, or has a mass of about 900 kilodaltons. It's L-shaped, and it's soluble with a globular arm, although we have the foot, if we call this a boot, or the L part, is embedded in that inner membrane. There are two faces here. There is the P face, or the positive side, and that is on the occurs on the in the inner membrane space, where those protons are pumped. And there is a negative face, which is the matrix side of that membrane. Names for this complex, should have started with that maybe. NADH coenzyme Q reductase, or NADH dehydrogenase. Both names are acceptable. You should probably recognize both of them. But basically its function is to oxidize NADH and reduce coenzyme Q. So first, we have NADH that has diffused from the citric acid cycle or the glycolysis, where it's produced, interacts or it binds to the enzyme on the matrix side of the inner mitochondrial membrane and transfers 2 electrons to FNM, our flavin mononucleotide. is reduced to FMNH2. FMNH2 then transfers 2 electrons, one at a time, to a second redox center, which is an iron-sulfur cluster. FMNH2 FMN has 3 oxidation states, I should say. FMNH2 is fully reduced. FMNH is referred to as a semiquinone, and FMN is fully oxidized. Finally, the iron-sulfur center transfers 2 electrons to ubiquinone, coenzyme Q, same thing. This is a mobile electron carrier. It can diffuse freely in the hydrophobic core of the inner mitochondrial membrane, and it can also shuttle electrons from complexes 1 and 2 to complex 3. Ubiquinone also has 3 oxidation states. UQ, or coenzyme Q, is fully oxidized. UQH, or QH, has picked up 1 electron, so it's a semiquinone, and then the fully reduced molecule is UQH2, or ubiquinol. Think of it this way: ubiquinone, known, no electrons; ubiquinol, all electrons. So it is fully reduced. Then finally, there is the transport of hydrogen ions into the cytosolic side of that inner membrane. The second complex we're going to look at is complex 2, and this is succinate dehydrogenase from the citric acid cycle. It's more like a tributary. It does not contribute to the proton gradient, so it doesn't directly contribute to the free energy of its oxidation, reduction to ATP synthesis, but it does feed reducing ubiquinol into, or reduced ubiquinol into the electron transport chain. This is the only citric acid protein or enzyme that is an integral membrane protein in the inner mitochondrial membrane. So as you see, we have succinate, which obviously has to diffuse to this complex, and it in the process of its conversion to fumarate, FAD is reduced to FADH2. So it picks up 2 electrons and 2 protons. 2 protons are released as it passes individually, 1 electron at a time, to an iron-sulfur center, and then we have the reduction, or contributes, I should say, to the reduction of ubiquinol. Now we're going to look at the third complex. The third complex is probably the most complex of all of the membrane embedded redox centers in that electron transport pathway. What occurs here is what's called the Q cycle, where ubiquinol, CoQH2 or UQH2, undergoes a 2-cycle reoxidation in which the semiquinone is a stable intermediate. At the same time, there's transport of protons into that inner membrane space. So before we discuss the transfer of electrons and the proton pumping that occurs in this complex, I'd like to point out that we're now seeing the cytochromes playing a role in this process. So cytochromes are proteins with heme prosthetic groups. They're responsible for the purplish brown color of the mitochondria. They're kind of purple because of that heme. Unlike heme groups and hemoglobin and myoglobin, which we've looked at, these groups undergo reversible 1 electron reductions from Fe3 plus to Fe2 plus. So in complex 3, we see both cytochrome C1 and cytochrome B sub L. So these are going to play important roles in this process. There are two phases. There's the first half of the Q cycle and the second. So we're going to look at these individually. Complex 3 has 2 active sites where the quinone cofactors undergo redox reactions. There's always a pool of UQH2 and UQ in that inner membrane. Remember, these are lipid-soluble proteins. So how does this work? UQH2 diffuses into the QP site. This is a two-step oxidation that it undergoes then. UQH2 is going to be reduced, or oxidized, I should say, to the semiquinone. So we have 2 protons being pumped into the inner membrane space, and then we have 1 electron being transferred to an iron sulfur center. From the iron sulfur center, that electron is transferred to cytochrome C1, and then finally out of the complex into that inner membrane space to cytochrome C. The second oxidation occurs and we get the quinone. No electrons, no protons. That diffuses back into the membrane. That electron is transferred to cytochrome B sub L, which subsequently, whoops, I missed this cytochrome, transfers that to cytochrome B sub H. In the second site, this can receive ubiquinone. So ubiquinone diffuses from the membrane into the second site and picks up an electron from cytochrome BH. This is the QN site. So now we have the semiquinone. This completes the first half of the Q cycle. Now let's take a look at the second. It's quite similar to the first half. A second molecule of UQH2 or co-QH2, however we want to represent it, enters that QP site. It is oxidized to the semiquinone and subsequently to the quinone. Again, 2 protons are pumped across that into the inner membrane space. We have 1 electron transfer to the iron sulfur center to site C1 to site C. Second electron transfers to site BL, or cytochrome BL, to site BH. And then again, from the first phase, we have the semiquinone. This picks up the second electron, is reduced to UQH2, which then diffuses out into the cytosol. So this part of the pathway, following the oxidation of a second UHQ, or UQH. Finally, we're going to look at complex 4, which also transfers electrons to cytochrome C and is also responsible for reduction of molecular oxygen. So we have the transfer of electrons to cytochrome C. This is a water-soluble molecule, so it can diffuse through the matrix to cytochrome or complex 4. In this particular membrane-embedded enzyme protein, we finally see our copper ions. So we have 2 that participate in this process. We have also a couple other cytochromes. And remember, we have two electrons being transferred in this process from complex 3. This complex contains 13 subunits in each half of a dimeric complex. So what do I mean by that? We have the dimers, we have the cytochromes, we also have the 2 copper ions. So electrons are then transferred to Cu1, and electrons are then going into the site A heme group. From site A, we get transfers to site to site A3, and then to CUB. Finally, CUB is responsible for the reduction of molecular oxygen to water. So this concludes the discussion of the complexes involved in the electron transport chain. Now we're going to look at what happens with that proton gradient that has been building up in that intermembrane space and the enzyme ATP synthase.